Hello, everyone. Welcome to another episode of Crafted Science, a place where science is crafted to explain the most complex scientific concepts for better understanding for grades 9 through 12 to ensure you're ready to take the next step into the collegiate level. Today, we will be discussing the cell cycle, cell signaling, and how they correspond to disease and cancer development. Let's dive into our lesson for today. First, we'll be talking about the cell phases. There are 5 phases within your cell: the G1 phase, G0 phase, S phase, G2 phase, and M phase. Your G1 phase is your first gap phase. This is where your cell grows physically larger, copies organelles, and makes the molecular building blocks it will need later in the cycle, such as mRNA and DNA polymerase and more. The G0 phase can be interchangeable, and it is also known as your resting state phase. All cells do not constantly divide, nor do they continue to go through the cell cycle. It is up to the body to tell which cells they want to replicate and which cells they want to stay put. The cells that normally do stay in the G0 phase are permanently stay there, and they just do their jobs, such as the neurons within your brain and nervous system. Next is your S phase. This is where your cell synthesizes a complete copy to the DNA in the nucleus. It also duplicates a microtubule organism structure called the centrosome. Centrosomes help separate DNA during the M phase. During the S phase, DNA polymerase is most present. It is responsible for the adding of the complementary base pairs of your DNA. And this is also where DNA replication occurs. Next is your G2 phase. During your G2 phase, this is also called the second gap phase. The cell grows more and makes proteins and organelles and begins to recognize its contents in preparation for mitosis. The G2 phase ends when mitosis begins. G1, S, and G2 phases are all known as interphase. During the meiotic phase or the M phase, your cell undergoes two division processes, one known as mitosis and the other known as cytokinesis. During mitosis, the nuclear DNA is condensed into chromosomes and the cell undergoes prophase, metaphase, anaphase, and telophase. Next, it undergoes cytokinesis, and that is the splitting of the cytoplasm into two daughter cells. Cytokinesis does not begin until mitosis ends. Next, we have our cell cycle checkpoints. Our cell cycle checkpoints are like the wardens of the cell cycle. We first have a checkpoint here at the G1 start checkpoint right here. This is what's looking for DNA damage. If any DNA damage is found within the cells, it automatically sends it down to the G0 phase. Here as well, apoptosis or cellular death will be induced for that damaged cell to be killed off. If it does pass the G1 checkpoint, it then enters the S phase for DNA replication and then into the G2 phase. Before you get to the meiotic phase, there's a G2 checkpoint. The G2 checkpoint checks on the cell size and makes sure all the growth factors are working correctly, and it also makes sure DNA replication happened correctly. It wants to make sure that nothing was replicated incorrectly and everything is in its respective base pair. If it isn't, the cell will be induced into apoptosis and cellular death. Lastly is our last checkpoint during the meiotic phase in between metaphase and anaphase of mitosis. This is called the spindle checkpoint. During metaphase and anaphase, your chromosomes line up in the middle, as we've researched and studied before, and they're pulled to their respective polar ends during anaphase. If the spindles do not form correctly and do not attach to the chromosomes correctly, they will not separate. Therefore, when it comes to the progression of the two daughter cells at the end of this cell cycle, you will not have the correct molecular components within the nucleus that you need to further go the signal that has been sent or undergo more cell cycles. Next are our wardens of the cell cycle. So what's keeping this going? What is making sure that everything is staying in its respective place? It is our cell cycle regulators. First, our cell cycle regulator we'll be talking about today is cyclins. There are 4 different cyclins that we have within a cell cycle. The first one is G1, G2S, S, and M cyclins. Each are in this respective cycle. They do not carry over into the next cycle. They progress and they lower in production when that prospective phase is over with. Cyclins are the most important core cell cycle regulator. In order for our cyclins to be able to do their job and progress the cell cycle along, it must have its best friends, our CDKs. CDKs naturally sit within the cell and they are inactive. They do not become active until a cyclin comes and connects with them and tells them, hey, come along, let's go walk these halls and make sure everything is good, everything's in its prospective place. These are the wardens of the cell cycle. Next, we have cell signaling. How do our signals talk to each other, and how are these signals sent to each other from our brains? There are 4 different types of cell signaling that our body undergoes. First is the paracrine, which is local, a cell that is closely related to each other that are able to talk. Next is our autocrine, which is self. Our cells themselves speak to each other and signal themselves. Endocrine is long distances, so this is if a cell within your arm is trying to get in contact with a cell in your foot, they send these distances through the endocrine system and they're through long distances. Next is direct contact, and this occurs through gap junctions, and this is from cells that are closely related or joined together. How are these signals sent and received to each cell? They're sent and received through our ligands and receptors. Ligands are the signaling molecules that your body sends out throughout the bloodstream, and it goes and tries to find its respective target or receptor on the cell that it needs to duplicate, replicate, or send the signal. That is our receptors, and that's our signal receiving protein. During our ligand and once our receptor bind to each other, an intracellular response happens, and this is where the response happens that we're looking for, like the cell to start the cell cycle process. So we all know cancer cells are a thing, and where do they come from? They come from the cell cycle and missed checkpoints and missed regulators. These are the few nitty-gritty mean boys that are able to pass through and create so many problems and troubles within our system. Cancer cells are they grow uncontrollably. The cell cycle regulators and normal signalings are turned off. So the checkpoints, they don't pass through. They're able to squeeze past them. Those are turned off. All of our regulators are turned off. So the cell is able to constantly regenerate itself. Next is to express telomerase. So telomerase is what is found at the top of each chromosome when it comes to duplication. Telomerase is what prolongs the life of cancer cells. So normally when your chromosome duplicates, the telomerase shortens, but it doesn't do this when it comes to cancer cells. It stays elongated, and this is what's able to keep the cancer cell going throughout the cell cycle and alive for much longer and to create these tumors. So within a lot of your genes, you have these things called tumor suppressor genes. So we're only going to talk about the main one because we're just hitting the target points. Everything you will dive way deeper in once you get to the college level. But right now, I want to focus on making sure you have the basis for understanding this cell cycle. So P53 is our most famous and primary tumor suppressor. They're mostly found at G1 checkpoint due to its job to halt the cell cycle and check the DNA to fix it or induce cellular death. Within cancer cells, P53 is often turned off or missing, allowing the damaged DNA to begin the replication process, thus centering 2 newly mutated cancer cells. No cancer cell is the same. They each have their own specific mutation. This is why cancer is so hard to treat, because once one cell is replicated and it enters that M1 phase and it passes that checkpoint, and enters into the S phase, it's no stopping it. It's going to constantly replicate, constantly duplicate, and cause new mutations each time a daughter cell is created. So if you think about it, this is how tumors form. This is why cancer cells constantly build and build and build on top of each other. They have no more room to grow but where they're building up at. And that's why the main treatment for cancer is radiation, because the radiation is able to go in and kill all of the surrounding cells, whether it's normal or abnormal. But the key thing is to make sure you're killing those mutated cancer cells so that they do not continue to replicate and proliferate throughout the body. So here are just a few diagrams. Just a normal cell. Everything, if it's unfixable, it goes through apoptosis because it hits that first checkpoint at the G1. And if not, cancer cells are able to continue to divide because it does not meet that checkpoint. Growth factors, P53, everything is turned off and it's able to keep going through the cycle. Here is just a better close-up of the telomerase I was talking about at the end of each chromosome, the part of the chromosome that does not shrink in cancer cells and which allows it to continue throughout the cell cycle and not undergo cell death. Here is just a diagram explaining normal P53 versus non-functional P53 and how it does not attach or send signals to our CDK and cyclins to come in and stop the process from going and induce apoptosis. It's able to keep going. Apoptosis is triggered. DNA replication is activated, and your cell doesn't halt at the G1 phase. The DNA is not repaired, and apoptosis is not triggered when your P53 does not bind to DNA and it's turned off or it isn't located within the cell at all. Cancer cells are able to manipulate your cells greatly. That's why it's such a deadly thing, and that's why cancer is so hard to pinpoint and treat, because once that signal is sent out and it passes that checkpoint, there's really nothing that you can do besides getting an abrasive form of treatment done, such as radiation or chemotherapy. It is amazing how your body is able to take out certain things and remove certain things despite the checkpoints and things that have been given. But like I said before, a lot of those things you will jump into deeply when you get to the collegiate level. But right now, I want you to pinpoint and make sure you guys know and are aware of the key points of the cell cycle, cell signaling, and how the abnormalities within cell cycle and cell signaling can lead to cancer cells and development of further death-related diseases. So just a conclusion, once again, we have our cell cycle of 5 different phases: our G0 phase, which is our resting phase; our G1 phase, which is our checkpoint phase, which is where we check for DNA damage; S phase is our DNA replication. S phase is our DNA replication phase. The G2 phase is where our body's cells are getting ready to undergo mitosis, and the M phase is where mitosis and cytokinesis happen, and that is where the two daughter cells are respectively created. Next, we have our cell cycle and our checkpoints. These are the G1, G2, and G2M and metaphase, anaphase checkpoints. These are just, once again, making sure we are finishing each phase correctly, making sure that there is no damage, making sure that the DNA has been replicated correctly, making sure that everything has undergone metaphase and anaphase correctly, so that we are not creating any known mutations or letting anything slip through. Then we have our wardens, our very last and crucial regulators of the cell cycle. We have our cyclins, which is what's producing in each different phase, and they have their own respective phase so that they do not topple over into the next person phase and cause us any problems. Then we have our CDKs, which is inactive until a cyclin comes into contact with it, and that is also known as CDKs are also known as kinases, and kinases are the enzymes that phosphorylates specific target proteins within our cells. Also, a regulator is our P53. That is a tumor suppressor gene. Our P53 comes in when the cell notices that DNA was not replicated correctly. Something got past a checkpoint, or it got past its respective phase without being apoptosed. So that's when P53 comes in. It pauses the cell cycle. It allows your body, if it wants to repair it, if it wants to start over, if it wants to just cancel the cell cycle altogether and induce apoptosis, it can and it will do that. That is also why P53 is so important when we jump down to cancer cells, because P53 is a sole regulator. It is our most important regulator, in my opinion. If that is not working, if it is absent in any type of way or malfunctioning, cancer cells can grow uncontrollably. They can be turned off. They can turn off other signals, other cell signaling and growth factors, which allows it to continue to grow, and which allows it to get past our other white blood cells that come and detect cells that need to be deleted or removed from our system. It's able to camouflage itself and put on normal traits to get past those other checkpoints and regulators, not within our cell cycle, but just within our system altogether. And lastly, the key thing with this is possessing telomerase, and that is that part of this chromosome that does not deteriorate, but it stays the same or grows